Today we'll be doing chapter 1 sexual reproduction in flowering plants. Let's start the reading of NCERT. Are we not lucky that plants reproduce sexually? The myriads of flowers that we enjoy gazing at, the scents and the perfume that we swoon over, the rich colors that attract us, are all there as an aid to sexual reproduction. Flowers do not exist only for us to be used for our own selfishness. All flowering plants show sexual reproduction. A look at the diversity of structures of the inflorescence flowers in the floral parts shows an amazing range of adaptations to ensure formation of the end products of sexual reproduction, the fruit and seeds. In this chapter, let us understand the morphology, structure, and the processes of sexual reproduction in flowering plants. Angiosperms. Flower, a fascinating organ of angiosperms. Human beings have had an intimate relationship with flowers since time immemorial. Flowers are objects of aesthetic, ornamental, social, religious, and cultural values. They've always been used as a symbol for conveying important human feelings such as love, affection, happiness, grief, mourning, etc. List at least 5 flowers of ornamental value that are commonly cultivated at homes and in the gardens. We do not need to read from here to here as this is not very important. Then we need to revise this diagram. Stigma, style, ovary, anther, filament, petal, and sepal. Pre-fertilization structures and events. Much before the actual flower is seen on a plant, the decision that the plant is going to flower has taken place. Several hormonal and structural changes are initiated which leads to the differentiation and further development of the flower primordium. Inflorescences are formed which bear the floral buds and the flower. In the flower, the male and female reproductive structures, the androecium and the gynoecium, differentiate and develop. You would recollect the androecium consists of a whorl of stamen representing the male reproductive organ and the gynoecium representing female reproductive organ. Here we need to remember that the androecium is the male reproductive organ's set and the gynoecium is the female reproductive organ set. Stamen, microsporangium, and pollen. Figure 1.2a shows 2 parts of typical stamen, the anther and the filament. The long slender stalk called the anther and the terminal, generally bilobed structure called the anther. The stalk is called the filament and the generally bilobed structure is called the anther. The proximal end of the filament is connected to the thalamus or the petal of the flower. The number and the length of stamen are variable in flowers of different species. If you were to collect stamen, each from 10 flowers, each from different species, and arrange them on a slide, you would be able to appreciate the large variation in size seen in nature. Carefully observation of each stamen under a dissecting microscope and making a neat diagram would elucidate the range in shape and attachment of anthers in different flowers. A typical angiosperm anther is bilobed with each lobe having 2 theca. Therefore, they are dithecous. So, an MCQ would come according to this, that the angiosperm anther is bilobed and it has 2 thecas, that means it is dithecous. It would also occur as how many thecas does an anther have. So you would write 4 thecas because each lobe has 2 thecas. Often a longitudinal groove runs lengthwise separating the thecas. Let us understand the variation. So here we have B part you could check out dithecous. Each lobe is dithecous and a longitudinal groove separates these thecas. Let us understand the various types of tissue and their organization in the transverse section of an anther. The bilobe nature of an anther is very distinct in the transverse section of the anther. The anther is four-sided, trigonal structure consisting of four sporangia located at the corner, two in each lobe. The microsporangia develop further and become pollen sacs. They extend longitudinally all through the length of the anther and are packed with pollen grains. So the anther is trigonal structure. It has four sides and consists of four microsporangia, 1, 2, 3 and 4, and each microsporangia later develops into a pollen sac, as you can see from the diagram, and each pollen sac extends longitudinally to the length of the anther, and these are packed with pollen grains. Structure of microsporangium. In a transverse section of a typical microsporangium appears near circular in outline. You would see here it is circular, but it's circular in shape. It is generally surrounded by four wall layers, which is epidermis, endothecium, middle layer, and the tapetum. And these layers are from outside to the inner. The outermost is the epidermis, then comes the endothecium, then comes the middle layer, and then comes the tapetum. The outer three walls perform the function of protection and help in the dehiscence of anther, which helps in release of the pollen grains or pollen, and the innermost wall, which is the tapetum, nourishes the developing pollen grains. So the cells of the tapetum possess a dense nucleus and generally are multinucleated. When the anther is young, a group of compactly arranged homogeneous cells called the sporogenous tissue occupies the center of each microsporangium. So the anther, which is usually very young, it is compactly arranged in sporogenous tissues, and it occupies the center of each center. This sporogenous tissue is like gives rise to these tissues, which are middle layer, epidermis, endothecium, and the tapetum. Then the sporogenous tissue later give rise to other things as well. So microsporogenesis. What is microsporogenesis? Is the process of formation of pollen grain. As the anther develops, the cells of sporogenous tissue undergo meiotic divisions to form microspore tetrads. जैसे जैसे एंथर डेवलप होता रहता है, the cells of the sporogenous tissue जो है वो microspore tetrad बनाता रहता है. So you could check it out over here. The A is the young anther, and here is the sporogenous tissue, which is surrounded by the layers, which is the epidermis, endodermis, middle layer, and the tapetum. The sporogenous tissue later give rise to microspore tetrad, which later give rise to pollen grains. And you could check it out that the middle layer is bilayered, not a single layer, and the tapetum is single layer, while the microspore mother cell later give rise to pollen grains over here. As each cell of the sporogenous tissue is capable of giving rise to a microspore tetrad, each one is a potential pollen or a microspore mother cell. The process of formation of pollen grain from pollen mother cell through meiosis is called microsporogenesis. So from here you could get assertion reasoning. That would be, the assertion would be each pollen grain is a potential microspore mother cell. So the reason would be each cell of the sporogenous tissue is capable of giving rise to a microspore tetrad. Then the microspores, as they are formed, are arranged in a cluster of four cells, the microspore tetrad. As the anthers mature and dehydrate, which is dehiscence happens, the microspores dissociate from each other and develop the pollen grain, develop into the pollen grains. So जैसे जैसे एंथर डेhiscence होता है, so pollen grain वैसे वैसे develop होता रहता है. And inside each microsporangium there are several thousands of microspora pollen grains that are formed that are released with the dehiscence of anther. Then later is the pollen grain. The pollen grains represent the male gametophytes. We need to remember this. This would be as an MCQ that what represents the male gametophyte, it is the pollen grain. If you touch the opened anthers of hibiscus or any other flower, you would find deposition of yellowish powdery pollen grains on your finger. Sprinkle these pollen grains on a drop of water taken on a glass slide and observe them under a microscope. You will be really amazed at the variety of architecture that you would see over here, which is the shape, sizes, colors, design. It would be very fascinating. So pollen grains generally, we need to remember the pollen grains are 25 to 50 micrometers in a diameter. Mu m would be the one that we would be given in the MCQ in diameter, and it has prominent two-layered. It is two-layered wall. It has two-layered wall. The outer one is exine, which is made up of sporopollen, which is the most resistant organic material known to human, and it can withstand high temperatures, strong acids or alkalis. And no enzyme is currently known to humans that it would degrade the sporopollen. And pollen grains, exine has a aperture called the germ pore. The sporopollen is actually absent. So what does it help in? So the germ pore helps in pollen tube formation actually. It is a guidance from where the pollen tube opens. Later, pollen grains are well preserved as fossils because of presence of sporopollen. We need to remember this as well. The question would come as presence of what makes the pollen grain preserved as a fossil. It would be sporopollen. And the other question is which layer of the pollen grain exhibits a fascinating array of pattern and design. It would be the exine. Then what do you think the exine should be? Why do you think the exine should be hard? What is the function of germ pore? The inner walls of the pollen grain is called the intine. It is a thin and continuous layer made up of cellulose and pectin. The cytoplasm of the pollen grain is surrounded by plasma membrane. When the pollen grain is mature, it contains two cells, vegetative and a generative cell, as shown here. And in figure 1.5b, the vegetative cell is bigger and has abundant food reserves and a large, irregular which is bigger, has abundant food reserve and has a large irregularly shaped nucleus. The generative cell is generally small and in the cytoplasm, and it is inside the cytoplasm of the vegetative cell. It is spindle shaped with dense cytoplasm and a nucleus, and in over 60% of the एंजियोस्पर्म पोलन ग्रेन जो है वो शेड होता है टू सेल्ड स्टेजेस में विच कंटेन्स वेजिटेटिव एंड जनरेटिव सेल, वाइल इन द रिमेनिंग स्पीशीज़ जो जनरेटिव सेल है वो दो मेल गेमीट्स में टर्न हो जाता है एंड माइटोटिकली एंड इट इज शेड एट 3 cell stage. So 40% 3 cell, 60% 2 cell. Pollen grains of many species causes severe allergies and bronchial afflictions in some people, often leading to chronic respiratory disorders such as asthma, bronchitis, etc. And it may be mentioned that Parthenium or carrot grass that came in India as a contaminant with imported wheat has become ubiquitous in occurrence and causes pollen allergy. Pollen grains are rich in nutrients. It has become a fashion in recent years to use pollen tablets as food supplements. In Western countries, a large number of pollen products in form of tablets and syrups are available in the market, and pollen consumption has claimed to increase the performance of athletes and racehorses. These are few examples of pollen products. When once they are shed, pollen grains have to land on stigma before they lose their viability if they have to bring about fertilization. So कितनी देर की हो सकता है viability? So it depends on, it is highly variable, and to some extent on prevailing temperature and humidity as well. कुछ सीरियल जैसे राइस एंड व्हीट में द वाइबिलिटी इज़ अबाउट 30 मिनट्स, while कुछ रोजेसी, लेग्यूमिनेसी एंड सोलेनेसी फैमिली में द वाइबिलिटी इज़ फॉर few months. You may have heard of storing semen or sperm of many animals, including human, for AI, which is artificial insemination. So it is also possible to store pollen grains of a large number of species for years in liquid nitrogen at about minus 196 degrees Celsius. Such stored pollens could be used as in pollen banks, similar to seed banks, in crop breeding programs. Next is the pistil megasporangium, which is ovule and embryo sac. जो gynoecium है, it represents the female reproductive part of the flower. The gynoecium may consist of a single pistil, which is monocapillary, or may consist of many pistils, which is multicapillary. When there are more than one pistils that are fused together, it is syncarpous, while they may be free, which is apocarpous. So from here we need to remember that multicapillary syncarpous pistil is of papaver, and the multicapillary apocarpous gynoecium or pistil is of michelia. So I have remembered it as AM, which means apocarpus michelia, and PS, papaver syncarpus. AMPS. Then each pistil has three parts: the sigma, style, and ovary. The sigma serves as the landing platform for pollen grains. The style is the elongated, slender part beneath the stigma, and the basal bulged part of the pistil is the ovary. Inside the ovary is the ovarian cavity, which is the locule. You need to remember that. Where is the ovarian cavity located, or what is the ovarian cavity called? It is the locule, and it is inside the ovary. The placenta is located inside the ovarian cavity, which is the locule. And in the previous class we studied about different types of placentations as well, and the definitions as well. And arising from the placenta are the megasporangia, commonly called the ovules. We need to remember that the placenta से arise करते हैं megasporangias, and they are called the ovules. जो number of ovules जो हैं per ovary, they may differ. That एक भी हो सकता है as in wheat, mango, paddy, and बहुत ज़्यादा भी हो सकता है as in papaya, watermelon, orchids. Then megasporangium ovule. Let us familiarize ourselves with the structure of typical angiosperm ovule. The ovule is a small structure attached to the placenta by means of a stalk called funicle. The body of the ovule fuses with the funicle in the region called hilum. Thus, one or two protective envelopes called integuments इसके पास होते हैं. So first of all हमें क्या रिमेंबर करना है कि ओव्यूल जो है वो छोटा सा स्ट्रक्चर होता है प्लस ओवेरियन कैविटी के अंदर, एंड इट इज़ अटैच्ड थ्रू फ्यूनिकल, एंड जहाँ पे फ्यूनिकल और ओव्यूल फ्यूज़ करते हैं, मीट करते हैं, उसे कहते हैं हाइलम. और उसके बाद जो है ओव्यूल वो इंटैगुमेंट से कवर्ड होता है, जो कि एक भी हो सकते हैं, दो भी हो सकते हैं. और जो इंटैगुमेंट है वो न्यूसेलस को इंसर्कल करता है एक्सेप्ट एट द माइक्रोपाइल रीजन. एंड जो ऑपोजिट ऑफ माइक्रोपाइल है दैट इज़ द चलाजलन और चलाजा, रिप्रेजेंटिंग द बेसल पार्ट ऑफ द ओव्यूल. Enclosed within the integument is a mass of cells called the nucellus. Cells of the nucellus have abundant preserved food materials. So integument के बीच में होता है nucellus, and cells of nucellus जो है उनके पास reserves of food reserves होते हैं abundant, and located in the nucellus is the embryo sac or the female gametophyte. You need to remember over here कि embryo sac को ही female gametophyte कहते हैं, पोलन ग्रेन्स को मेल गेमेटोफाइट कहते हैं. An ovule generally has single embryo sac formed from a megaspore. So now let's go to the megasporogenesis. The process of formation of megaspores from a megaspore mother cell is called megasporogenesis. And ovules generally differentiate a single MMC, which is megaspore mother cell, in a micropylar region of the nucellus. It is a large cell containing dense cytoplasm, a prominent nucleus. The MMC undergoes meiotic division, and meiosis results in the formation of 4 megaspores. So let's get to it. ये देखो, ये है हमारे पास nucella. This is the micropylan, this is the chalazoland, and this is the megaspore mother cell over here. It contains the nucleus over here. Now the division has taken place. We have 4 megaspore tetrad, out of which only one remains while the other 3 degenerates. So female gametophyte, which is the ovule or the embryo sac, is the female gametophyte. In a majority of flowering plants, one of the megaspore is functional while the other 3 degenerate. Only the functional megaspore develops into the female gametophyte, which is the embryo sac. This method of embryo sac formation from a single megaspore is termed as monosporic development. This is a question that would come in the anywhere it could come. The method of embryo sac formation from a single megaspore mother cell or a single megaspore is termed as monosporic development. What would be the ploidy of the cells of nucleus, nucellus, MMC, and the functional megaspore and the female gametophyte? So these 3 would have the polarity N, while nucellus would have a polarity of 2N, because MMC has 2M, because this would later go on into meiotic division, and nucellus is food reserves, which does not go. The functional megaspore and the female gametophyte are employed. Let's check it once. What is the ploidy of functional? So ploidy N, yeah, it is N. We could remember that as well. Now the nucleus of the megaspore divides. Nucleus of the functional megaspore divides mitotically to form 2 nuclei, which moves to the opposite poles, forming the 2 nucleate embryo sac, later 4, then 8 stages. And it is of the interest to note that these mitotic division are strictly free nuclei, which means that the nuclear divisions are not followed immediately by cell wall formation. After the 8 nucleate stages or the stage, cell walls are laid down by cell wall formation. This leads to the formation of female gametophyte called the embryo sac, and we should observe the distribution of cells inside the embryo sac, which is 6 out of the 8 nuclei are surrounded by cell walls and organized into the cell, and the remaining 2 nuclei called the polar nuclei are situated below the egg apparatus, and these are called whole cell is called the central cell. So this is the structure, this is the antipodal cells, this is the synergid and the egg cell. This whole, the synergid and the egg cell, is called the egg apparatus. This is the central cell, polar nuclei, and these are the antipodals. There is a characteristic distribution of the cells within the embryo sac. 3 cells are grouped together at the micropylar end, which is the top end, and constitute the egg apparatus. The egg apparatus contains 2 synergids and 1 egg cell. And the synergids have a cellular thickening at the micropilot tip called the filiform apparatus, which plays the important role into guiding the pollen tube into the synergids. And the three cells at the chalazal end are called the antipodal cells, and the large central cell, as mentioned earlier, has two polar nuclei. Thus, a typical angiosperm has 8 nuclei and 7 cells. So we need to remember here in the diagram, this is the chalazal end while this is the micropylar end, and over here in the above diagram the top one was the micropylar. Pollination. In the preceding sections you have learned that the female and male gametes in the flowering plants are produced in the pollen grain and embryo sac respectively. As both types of the gametes are non-motile, they have to be brought together for fertilization to occur. How is this achieved? Pollination is the mechanism to achieve this objective. Transfer of pollen grains to the stigma of a style is called pollination. Flowering plants have evolved an amazing array of adaptation to achieve pollination. They make use of external agents to achieve pollination. So external agents could be wind, animal, water, etc. And kind of pollinations are, there are three kinds of pollination depending on the source of pollen. First of all, it is autogamy. In this type of pollination is achieved within the flower, within the same flower actually, and transfer of pollen grains from anther to stigma of the same flower. In a normal flower which opens and exposes the anther and the stigma are completely autogamy, which is rather rare. The complete autogamy is actually very rare. The autogamy in such flowers requires synchrony in pollen release and stigma recessivity as well, and the anther and the stigma should be very close to each other for cell pollination to occur. Some examples are viola. Some examples which has chasmogamous flowers and clistogamous flowers are viola, oxalis, and commelina. Viola is also called common pansy, and we could remember this by COV. I have remembered COV through COVID. COV, commelina, oxalis, and viola. So there are two types of flowers they produce: commelina, oxalis, and viola, which is the chasmogamous flowers which open like regular flowers with exposed stigma and anther, while there is another clistogamous flowers which do not open at all. So this is the example of clistogamous flowers. This is an example of common pansy, I guess, viola, and this is the example of cross-pollinated sunflower. In such flowers, the clistogamous flowers, the anther and stigma lie very close to each other, and when the anther descends in the flower buds, pollen grain come into contact with stigma to effect pollination. Thus, clistogamous flowers are invariably autogamous, as there is no chance of cross-pollination landing on the stigma, and we need to remember that the clistogamous flowers produce a short seed set even in the absence of pollinators. Then we have ketonogamy, which is the transfer of pollen grains of the anther from the anther to the stigma of another flower of the same plant. Ketonogamy is functionally cross-pollination, while genetically it is autogamy. Xenogamy is the transfer of pollen grains from anther to stigma of different plant, and this is the only type of pollination which during pollination brings genetically and functionally different types of pollen grains to the stigma. Then we have agents of pollination, which plants usually have two: abiotic, which is wind and water, and one biotic, which is animal insects, to achieve pollination. Majority of plants use biotic agents for pollination. Only a small proportion of plants use abiotic pollinators, and pollen grains coming in contact with the stigma is a chance factor in both wind and water pollination. To compensate for this uncertainties and associate lots of pollen grains, these flowers produce enormous amount of pollen when compared to the number of ovules available for pollination. So we need to remember that in wind and water pollinated flowers, The flowers produce enormous amount of pollen when compared to the number of ovules available for pollination. Then we have pollination by wind. Pollination by wind is more common amongst abiotic pollination. Then wind pollination requires, we need to remember this, light non-sticky pollen grains so that they can be transported in the wind. They have well exposed stamens as that so that the pollens are easily dispersed into wind currents, and large, often feathery stigma to easily trap airborne pollen grains. Wind pollinated flowers usually have single ovule in each ovary and many flowers. Numerous flowers are packed into an inflorescence. An example is tassel of a corn cob. You see, are nothing but the stigma and style which wave in the wind to trap pollen grains. Wind pollination is quite common in grasses. Now, pollination of water is quite rare. We need to remember that, and it is limited to 30 genera of monocotyledons, mostly monocotyledons. As against this, you would recall that water is a regular mode of transport for male gametes among the lower plant groups such as algae, bryophytes, and pteridophytes. It is believed, particularly for some bryophytes and pteridophytes, that their distribution is limited because of the need of water for transport of male and female gametes and fertilization. Some examples are Vallisneria and Hydrilla, which are freshwater, while Zostera, which is seagrass, is marine. We need to remember over here that Vallisneria, Hydrilla are freshwater, water pollinated plants, while Zostera is a marine. Not all aquatic plants use water for pollination. In majority of aquatic plants such as water hyacinth and water lily, the flowers emerge above the level of water, and these are later pollinated by insects or wind. So here we need to remember that amongst water lily and water hyacinth, the plant is actually pollinated through wind or insects. In Vallisneria, which is a freshwater plant, the female flower जो है वो emerges at the surface of water, while the male one is emerged inside the water or to the just below the surface of water, and it releases the male gametes or the pollen grain, and then they are carried passively through water currents, and eventually they are pollinated. Then we have another group of water pollinated, which is seagrasses. Example Zostera, that the pollen grains are released inside the water and they are pollinated. And you need to remember that pollen grains are generally long, ribbon-like, and carried passively inside the water to achieve pollination. Then some of them reach the stigma and achieve pollination. In most of the water pollinated species, pollen grains are protected from wetting by mucilaginous covering. We need to remember that as well. In water pollinated species, mucilaginous covering is actually present. Both wind and water pollinated flowers are not very colorful and do not produce nectar because they do not want to attract these insects or animals. This is the reason why. Then majority of flowering plants use a range of animals for pollination. Examples: bees, butterflies, flies, beetle, wasp, ant, moths, birds, etc. Then among the animal insects, particularly bees are very important pollinating agents. Uh, Among the bees are dominant biotic pollinating agents. Even large animals such as primates like lemur, arboreal rodents which are tree dwelling, which means they are living on the trees, or even reptiles as gecko lizard, garden lizard, are pollinators in some species. Often, flowers of animal pollinated plants are specifically adapted for a particular species of animal. जो फ्लावर्स है एनिमल पॉलिनेटिंग वाली वो कुछ स्पीशीज़ के लिए अडेप्टेड होती है बिकॉज few produce smell, fragrance actually, and few produce bad essence, or you could say bad smell, to attract desired animals. Majority of insect pollinated flowers are large, colorful, fragrant, and rich in nectar. Whilst when the flowers are small, a number of flowers are clustered into inflorescence to make them conspicuous, and animals are attracted to this by fragrance or colors. The flowers pollinated by flies and beetles secrete foul odors to attract these animals. To sustain animal visits, flowers are to provide rewards to them, such as nectar or pollen grain. For harvesting these rewards from the flower, the animal visitor comes in contact with the anther and the stigma. The body of the animal gets a coating of the pollen grain, which are generally sticky in animal pollinated flowers, and when the animal carrying the pollen on its body comes in contact with the stigma, it brings about pollination. Now, in some species of floral, what are example are the safe place to lay eggs, as an example of Morphilus, which is the tallest plant flower, and Amorphilus is about 6 feet in height. And similar would be between yucca and moth, a similar relationship in which yucca brings about pollination. Moth brings about pollination, while the yucca provides a safe place for moth to lay eggs. The larva comes out when the seed starts developing. So locule was the ovarian cavity. So the moth deposits its egg in the ovarian cavity. Then we don't need to read this. Then outbreeding devices. Majority of flowering plants produce hermaphrodite flowers, and pollen grains are likely to come in contact with the stigma of same flower. Hermaphrodite means both the sexes are present on the same flower, and continued self-pollination results in inbreeding depression, which means there are no variations, no resistance to any new disease. And flowering plants have developed many devices to discourage self-pollination and to encourage cross-pollination. In some species, pollen release and stigma receptivity are not synchronized, which means कि जो pollen grain जब release होगा, descent of anther जो होगा वो स्टिग्मा रिसेप्टिविटी मतलब स्टिग्मा जब पोलन ग्रेन को रिसीव करने के लिए रेडी होगा वो सिंक्रोनाइज मतलब साइमल्टेनियस नहीं होगा, वो पहले कुछ होगा दूसरा कुछ होगा. Either the pollen grain is released before the stigma receptivity. In some other species, the anther and stigma are placed at different positions so that the pollen do not comes in contact with the stigma of the same flowers. Both these devices prevent autogamy, not geitonogamy. Okay? Then the third device to prevent inbreeding is self-incompatibility, which prevents getono as well as autogamy. So the third device to prevent inbreeding is self-incompatibility. This is a genetic mechanism and prevents self-pollen from fertilizing the ovules by inhibiting pollen germination or pollen tube growth in the pistil. Another device to prevent self-pollination is production of unisexual flowers. Both female and male flowers are present in the same plant, such as castor maize. We need to remember that castor maize are monoecious. It prevents autogamy but not geitonogamy. In several species such as papaya, male and female flower present on different plants. So that is Dioichthys dioichi, which is unisexual flower, you could say. And presence of this kind of flower where female or male parts are present on different plants, it prevents autogamy or geitonogamy. Pollen pistil interaction. Pollination does not guarantee the transfer of right type pollen, which is compatible pollen of the same species as the stigma. Often pollen of the wrong type, either from the other species or from the same plant, which is self-incompatible, also lands on the stigma. The pistil's ability to recognize these pollen and whether it is of the right type or the wrong type. If it is of the right type, the pistil accepts the pollen and promotes post-pollination pollination events, that is leads to fertilization. If the pollen is of the wrong type, the pistil rejects the pollen by preventing pollen germination on the stigma or the pollen tube, which is growth in the style. This ability to recognize the pollen followed by its acceptance or rejection is the result of continuous dialogue between the pollen grain and the pistil, and this dialogue is mediated by chemical components of the pollen interacting with those of the pistil. It is the only recent years that botanists have able to identify some of the pollen and pistil component and the interaction leading to recognition followed by acceptance or rejection. Here we need to remember that is the pollen grains that are germinating on the stigma. B is the growth of pollen tube through the style, and C is the longitudinal dissection of the pistil showing the path of the pollen tube growth. This is the D1. Here is the enlarged view of the egg apparatus, which contains the egg cell and the two synergids, showing the entry of the pollen tube into the synergid. Then E1 is the discharge of the male gametes. This is the male gamete 1, this is the male gamete 2 into the synergid, and the movement of sperms, one into the egg and the other into the central cell. So this one is going into the central, while this is the synergid, and this male gamete is going to the egg cell. As mentioned earlier, following compatible pollination, the pollen grain germinates on the stigma to produce a pollen tube through one of the germ pores. The contents of the pollen grain move into the pollen tube. The pollen tube grows through the tissue of the stigma and style reaches the ovary. You would recall that some plants pollen grains shed at two-cell condition, which is 60% of angiosperms. In such plants, the generative cell divides to form two male gametes during the growth of pollen tube in stigma. In plants which shed pollen in three-cell condition, pollen tube carry two male gametes from the beginning, which was 40% of the angiosperm. Pollen tube, after reaching the ovary, enters the ovule through the micropyle and enters one of the synergids through the filiform apparatus. Many recent studies have shown that the filiform apparatus present at the micropylar part of the synergids guide the entry of the pollen tube. All the events from deposition of pollen grains, deposition of pollen grains on the stigma, until the pollen tube enters the ovule are referred to as pollen pistil interaction. This is a 3-marker to 5-marker question that would come that explain the pollen pistil interaction and other things as well. So you need to summarize all these things, that is right type, wrong type, then pollen tube entry and entry of pollen tube to the synergids, everything, and filiform apparatus, what is filiform apparatus, etc. As pointed out earlier, pollen pistil interaction is a dynamic process involving pollen recognition followed by promotion or inhibition of pollen. So we need to remember this for MCQ point of view, that pollen pistil interaction is a dynamic process which involves pollen recognition, promotion or inhibition of the pollen. So this would come as an MCQ. Uh, then the knowledge gained in this area would help the plant breeder in manipulating pollen crystal interactions even in incompatible pollination to get desired hybrid. You can easily study pollen germination by dusting some pollen flowers, pollen grains from flowers such as pea, chickpea, etc. on glass side containing drop of sugar which is 10%. After about 15 to 30 minutes, observe the slide in the low power lens of the microscope. You are likely to see pollen tubes coming out of the pollen grains. A breeder is actually interested in crossing different species and often genera to combine desirable characters to produce commercially superior varieties called hybrids. And artificial hybridization is one of the major approaches of crop improvement program. In such crossing experiment, it is important to make sure that only the desired pollen grains are used for pollination and the stigma is protected from contamination. So this is done by bagging and emasculation. What is bagging and what's emasculation? If the female parent bears bisexual flowers, the removal of anther from the flower bud before the anther dehiscence using a pair of forceps is necessary. This is called emasculation. And then the emasculated flower has to be covered using a bag which is called bagging. And the bag is usually made up of butter paper and it prevents the entry of unwanted pollen. When the stigma of the bag flower attains receptivity, then the pollen grains of the desired parent, male parent, is listed on the stigma and the flower is rebagged and the fruit is allowed to develop. If the flower female parent produces unisexual flower, there is no need of emasculation. Actually, if the female parent produces unisexual flower, which means only one sex is present, there is no need of emasculation. The female flowers are bagged before the flowers open. Once the stigma becomes receptive, pollination is carried out using desired pollen and the flowers is rebagged. Next is double fertilization. After entering one of the synergids, the pollen tube releases two male gametes into the cytoplasm of the synergids. One of the male gametes moves towards the egg cell and fuses with the nucleus, thus completing syngamy. We need to remember this. The other one, and this results in the formation of zygote. Syngamy results in the formation of zygote. The other male gamete moves towards the two polar nuclei located in the central cell and fuses with them to form triploid PEN, which is primary endosperm nucleus, which later forms PEC, which is primary endosperm cell. So we need to first remember that PEN is formed, later PEC is formed. This is a MCQ that we could get in NEET or in CBSE. As this is the fusion of three haploid nucleus, this is called triple fusion, and as two fusions are involved, which is syngamy and triple fusion takes place in the embryo sac, the event is called double fertilization, and it is unique to flowering plants, which is angiosperm. The central cell after the triple fusion becomes PEC and develops into the embryo sac, and the zygote later develops into the embryo. And first of all, endosperm formation takes place, which means first of all, if the whole process needs to take place, first of all, the formation of PEN, then zygote, then PEN to PEC and endosperm, and then from zygote to embryo. We need to remember this as well. This is another page. Then forced fertilization structures everywhere. Following double fertilization events of endosperm and embryo development, maturation of ovule into seeds and ovary into fruits are collectively termed post-fertilization events. Endosperm development precedes embryo development. Why? The primary endosperm cell divides repeatedly and forms diploid endosperm tissue. You need to remember diploid. 3N is uh, 3N is the diploidy level of the endosperm tissue. The cells of the tissue are filled with food reserves and are used for nourishment or nutrition uh, of the growing embryo or the zygote, I would say. In most common types of endosperm development, the PEN undergoes successive nuclear division, giving rise to few nuclei. The stage of endosperm is free nuclear endosperm, and the cell formation occurs, and the endosperm becomes cellular. The number of free nuclear formed before cellularization varies greatly, actually, and the coconut water formed from the tender coconut is actually free nuclear, and the surrounding white kernel is actually cellular endosperm. The endosperm may either be completely consumed by the developing embryo, as in pea, groundnut, castor beans, or it may persist in mature seed, as in castor and coconut, and could be used for seed germination as well. Then embryo develops into at the micropyle end of the embryo sac where the zygote is situated. Most zygotes divide only after a certain amount of endosperm is formed. We need to remember this. This is an adaptation to provide assured nutrition for the developing embryo. This is an assertion reason. Though the seeds differ greatly, the early stages of the embryo development embryology are similar in both monocots and the dicots. Figure 1.3 depicts the stage of embryogeny and in dicot embryo, and the digot gives rise to first pro-embryo, then globular, then heart-shaped, and then mature. We need to remember this. This is the pro-embryo, which is not the pro-embryo, which is actually the cell where after the fertilization, actually double fertilization. This is the zygote, and this you could get the pro-embryo. This is the globular embryo, and which forms a heart-shaped embryo. And we need to remember that these are the suspensor cells. Then this forms actually the seed, and here it is the mature embryo. This is the suspensor, this is the radicle, this is the cotyledons, and this is the plumule. A typical dicot embryo consists of a embryonal axis and two cotyledons. This is the MCQ or fill in the blanks that a typical dicot consists of an embryonal axis and two cotyledons. The portion of the embryonal axis above the cotyledon is epicotyle, which terminates with the plumule and or tip stem. So here we go. This is the cotyledon, which above is the plumule, and this is the below the region, which is hypocotyle, which terminates into radicle. The cylindrical portion below the level of cotyledon is called hypocotyle and terminates into lobe and called the radicle or the root tip, and the root tip is covered by root cap as well. The embryo of the monocot possesses only one cotyledon. In grass family, the cotyledon is called scutellum, which is situated at the lateral side of the embryonical axis. At the lower end, the embryonical axis has the radicle or the root cap enclosed in undifferentiated seed called the cleorhiza. First of all, cleorhiza comes here, then the radicle and the root cap, and the portion of embryonical axis above the level of the attachment of scutellum or the cotyledon contains the cleoptile, which contains the shoot apex called the plumule, and we also have an epiblast over here. We need to remember this. And the epicotyle has a shoot apex and a leaf primordial enclosed in the hollow follicle structure, the cleoptile. Then we could observe, then comes the seed. In angiosperm, the seeds is the final product of sexual reproduction. We need to remember this, where it comes as the MCQ that what is the final product of sexual reproduction? It is the seeds. It is often described as a fertilized ovule. Seeds are formed inside fruits. Remember this. Seed typically consists of seed coat, cotyledon, in seed typically consists of seed coat, cotyledon, and embryo axis. You need to remember this: seed coat, cotyledon, embryo axis. This you could remember as it was given above as well, that a typical seed consists of dicot embryo consists of embryonic axis and cotyledon. Similarly, cotyledon and embryo axis, and seed coat is for seed. Then the cotyledons of the embryo are simple structure, generally thick, swollen due to food reserves. So we could remember that as well as in legumes. The cotyledons जो होते हैं embryo में, वो simple structure होते हैं, वो thick होते हैं, swollen होते हैं due to food reserve की storage में, and कुछ जो होते हैं वो non और exalbuminous होते हैं, which means उनके पास कोई एंडोस्पर्म नहीं रहता. Example pea, groundnut and beans. And then we have कुछ में रह जाता है albuminous seed जिसे कहते हैं, as in wheat, castor, barley, maize. and कुछ में nucellus भी रह जाता है, which are perispermic seeds, and it is beetroot and black pepper. Here we had studied that. Here we go. Completely cumin, pea, groundnut, pea, और कुछ में प्रसिद्ध रह जाता है coconut, castor. These are important examples for meat point of view. Integuments of ovule hardens as a tough protective seed coat. Tough protective seed coat के जैसे these integuments harden, and the micropyle remains as a small pore in the seed coat. This facilitates the entry of oxygen and water into the seed during germination. You need to remember the importance of the small pore, which is the micropyle pore on the seed coat. What is its use? It is used for the entry of oxygen and water during the germination. As the seed matures, its water content is reduced and its seed becomes relatively dry, which is 10 to 15% moisture by mass. The metabolic rate activity embryo की वो भी slow down हो जाती है. So these could be asked in for 3 markers in CBSE, that what is the after seed formation activities. So first of all जो इंटेगुमेंट्स हार्डन हो जाती है, माइक्रोपाइल वाला लिख देंगे कि इज़ द पोर, एंड उसमें पानी और ऑक्सीजन के एंट्री के लिए वो पोर रहता है ऑन दी सीड कोट. सेकेंड है कि वाटर कंटेंट रिड्यूस हो जाता है, विच इज़ 10 to 15% बाय मॉइस्चर बाय मास. एंड मेटाबॉलिक एक्टिविटीज़ जो है वो स्लो डाउन हो जाती हैं, एंड एम्ब्रियो जो है वो डॉर्मेंसी में एंटर कर जाता है. एंड जब फेवरेबल कंडीशंस आ जाती हैं, then they germinate. It is important. As ovules mature into seeds, the ovary develops into a fruit. That is, the transformation of ovules into seeds and ovary into fruits proceeds simultaneously. The walls of the ovary develops into the walls of fruit called pericarp. You need to remember that जो ओवरी की वॉल है वो क्या बन जाती है, दैट इज़ द पेरिकार्प, विच इज़ द फ्लोर ऑफ फ्रूट. The fruit may be fleshy as in guava, mango, oranges, or it may be dry as in groundnut and mustard. You need to remember that. This is important for neat point of view as well as MCQs and CBSE. And many fruits have evolved mechanism of seed dispersal of seeds. As we call the classification of fruits and dispersal mechanism that we have studied in early classes in grade 11 and 9, 10 as well. Now we have in most plants, by the time fruit develops from the ovary, other floral parts degenerate and fall off. But some species, as in apple, strawberry, cashews, etc., the thalamus also contribution in the formation of seeds, and these are called false fruits. Most fruits, however, develop only from the ovary and are called true fruits. Although in most of the species, fruits are results of fertilization, there are a few species in which fruit develops without fertilization, and these are called parthenocarpic fruits. And banana, tomato, are such examples, and they are seedless. We need to remember here, this is the, you could say, the seed examples of seed over here, and structure of some seeds. We could remember these, we could not. For NEET point of view, it is very important to remember that this is cotyledon, this is microbilome, then this is the seed coat, this is the endosperm, these are the cotyledons, and these are the root axis or the hypocotyle. This is the shoot apical meristem, which is the blue over here. Then we could remember these. This is the single one, monocots, and this is the cotyledon, this is the shoot apex, this is the root axis and the root tip, and this is the endosperm. This is again the other structure of the seed, mature seed, which is the endosperm, the pericarp, and the scutellum, cleoptile, chlorhiza, root apex, the radicle, and the plumule. If you get a diagram, to draw the diagram between for these, you could either draw this one, which is, I find this is a bit easier than this one. I find that one much easier than this. You could draw any of these. Seeds offer several advantages to angiosperm. Firstly, what are the advantages of seeds? Since reproductive processes such as pollination, fertilization are independent of water, the seed formation is actually more dependable. Also, seeds have adaptive strategies for dispersal or to new habitats and help the species to colonize in other areas. Also, they have sufficient food reserves, and young seedlings are nourished until they are capable of photosynthesis on their own, and the hard seed coat protects the young embryo, and they could be they after the sexual reproduction they could form new genetic combination leading to variations as well. Now, seed is the basis of agriculture. Dehydration and dormancy of mature seeds are crucial for storage of seeds, which can be used as food throughout the year and also to raise crop in the next season. Can you imagine agriculture in the absence of seeds or in the presence of seeds which germinate straight away soon after formation and cannot be stored? नहीं हो सकता ऐसा. How long do seeds remain alive after they are dispersed? This period again varies greatly. कुछ स्पीशीज़ में वो कुछ मंथ्स रहता है, कुछ सालों के लिए भी रहता है, कुछ हंड्रेड्स ऑफ ईयर के लिए भी. एग्जाम्पल है लिपाइनस आर्टिकस, जो कि हमने आर्टिक टुंड्रा से फाइंड किया था. इट हैड 10,000 ईयर्स ऑफ डोरमेंसी. एंड रीनिक्स है, रिसेंट वाला है फीनिक्स डेक्टाइलिफेरा, एंड विच इज़ द डेट पाम ट्री फ्रॉम द किंग हैरोल्ड प्लेस. Now, ये important है for both NEET and this one is important for NEET point of view as well as other. So we need to remember the Lupinus Arcticus का 10,000 years का था for dormancy, and it was found in Arctic tundra, while Phoenix Detylifera का 2,000 years का dormancy period, which was found in King Harold's palace near the Dead Sea. After completing a brief account of sexual reproduction of flowering plants, it would be worth noting or worth attempting to comprehend the enormous reproductive capacity of some flowering plants by asking the following questions. How many eggs are present in an embryo sac? How many embryo sacs are present in an ovule? And how many ovules are present in ovary? ये depend करता है accordingly. Can you think of some plants in which fruits contain very large number of seeds? Orchids, then papaya, watermelon. Orchids plants are such category in which each fruit contains thousands of tiny seeds. Similarly is the case of fruits from parasitic species Orobranchi and Striga. These are important ones that you need to remember. Orobranchians striga, orchid fruits. These contain many, and the question could be asked accordingly that कि कौन से parasitic species में multiple seeds होते हैं. Now, apomixis and polyembryony. All the seeds in general are products of fertilization, लेकिन कुछ species में जैसे Asteraceae, which is the grass family, and grasses, वैसे दोनों ही लिखते थे Asteraceae and grass, that would have been the same thing, have evolved a special mechanism to produce seeds without fertilization, that is the apomixis. So we need to remember the definition, that is the formation of seeds without fertilization is called apomixis. Now, what is fruit production without fertilization called? That is the parthenocarpy. I would say, thus apomixis is a that we had here that fruits develop without fertilization, parthenocarpy, and thus apomixis is a form of sexual reproduction that mimics sexual reproduction. There are several ways that developing the apomictic seed. In some species जो diploid egg cell था वो formed होता है without the reduction division, which is meiotic, जो हमें पहले पता था था that is meiotic division, that does not division from 4 cells, and later से एक बचता है, then वो divide होता है mitotically, फिर fertilization होती है, then diploid वो
कितनी दे चलनी आ तेरी मम्मी अस्पताल गई हुई है बस ओदे ध्यान रखी अच्छा.
More often, as in many citrus and mango varieties, some of the nuclear cells surrounding the embryo sac starts dividing to move into the embryo sac and develop into embryos. In some species, each ovule contains many embryos. Occurrence of more than one embryo in a seed is referred to as polyembryony. सबसे पहले तो let us get to know what is apomixis. Apomixis होता है formation of seed without reproduction. उसकी एक एग्जाम्पल दी है. उसके बहुत सारी फैमिलीज हैं. एक फैमिली है which does apomixis, that is grass family called Asteraceae, and एग्जाम्पल कैसे वो करता है, it is कि diploid egg cell ही बन जाता है without MMC going under reductional division, meiotical division, and वो embryo में later बदल जाता है without fertilization. फिर citrus and mango family में मैंगो वैराइटीज़ में जो न्यूसेलस सेल है वो प्रोट्रूड होने जाता है अंदर. सम ऑफ द न्यूसेलस सेल्स सराउंडिंग द एम्ब्रियो सैक start dividing and produce into the embryo sac and develops in the embryo and contains many embryo. It contains many embryo, and occurrence of more than one embryo in a seed is referred to as polyembryony. Now अगर हम take out some seeds and ऑरेंज के हम सीड्स को लेंगे और उसे स्क्वीज करेंगे, then बहुत सारे एम्ब्रियो दिखेंगे हमें of different shapes and sizes from each seed, and we could get to know. Hybrid variety of several of our food and vegetable crops are being extensively cultivated. Cultivation of hybrids have tremendously increased the productivity. One of the main problems of hybrids is that the hybrid seeds have to be produced every year, which means they could be very expensive. And अगर hybrids को हम apomixis के through बनाएंगे, then there would be no segregation of the characters in the hybrid progeny, and the farmers could keep these seeds to raise new crops each year. Hence, वो cheap पड़ेगा. So, importance of apomixis is very much in hybrid seed industry these years, and research going on here for genetics of apomixis and to transfer apomictic genes into hybrid varieties. So, हमारा यहाँ पे first chapter complete होता है fully. Do these questions as well, read कर लो, करनी नहीं है तो. Thank you.